I thank you, Elizabeth, for inviting me, and also Anne introducing my talk. Okay, this morning I want to present our database about sex description of the cells sold by commercial vendors. Okay, in January 2013, US FDA made an announcement that manufacturers of the drug Zolpidem should reduce the regular dose of at the time to half for women, because in clinical studies they found that after 8 hours of taking the drug of regular dose, women were 5 times more likely not to be awake compared to men. And it is, of course, causing a lot of accidents or impair their functions for their jobs and everyday life. Well, as you know, drug development process is a very long process and very costly. And it involves the developmental stage where people identify the compounds which has biological activity, and they are tested for the various ways how they are acting, and also whether they are safe, where there's no adverse safe. So when the compound has enough data to show that they have beneficial biological activity but no serious adverse effects, then they are given permission to test in humans, and then later to be marketed as a drug. So in preclinical phases, we do many experiments, and some studies are done in cells and some are done in animals before they are tested for human studies. Okay. So we all know now that it is important to know sex of your research subjects. Okay? So in humans and whole animals, it's relatively easy to identify the sex. So many studies, they don't report the sex, but it's just they are not careful enough to report. Or many studies were done in male animals, we know that. So now we have to do more experiments with females to get enough data. But what about cells? Do we know the sex of cells that we are doing the experiments? Of course, cells have chromosomes, and it has XX and XY, as very well explained by Barbara. But it's not easy for the researchers to know the sex of the cells. Okay. Does it matter whether your cells are male or female? Of course, yes. And it's not said by me, but it's said by Elizabeth Pollitter in 2013 in Nature. So many biological mechanisms and then pathways are using in the culture of the cells often the basis of the many development of drugs and then diagnostic and therapeutic interventions. But if you don't know whether it's from male cells or female cells, or you have equal data from both sexes, you are putting many people in danger, especially women, because most of the animal cells at the next stage are done in male animals. So if we see the literature, sex influences in stem cells and some primary cells are reported in the papers. So, but most of the preclinical studies, when you cell experiments, cell lines are used. And then many of the literatures do not report the sex of the cell lines they are using. So in a study by Taylor et al., which was published in Biology of Sex Difference, they looked at the studies from the 10 leading cardiovascular disease journals, and they found 50 to 100%, more than 70, 80%, did not report the sex of cells used in these studies. And none of those studies, 101 studies they looked into use of cells, only from female cells. So there were studies done in male cells, and there were studies done in unknown sex cells, but there was no, only female studies, female animal source was not reported. And in another study published in American Journal of Physiology, Cell Physiology in 2014, they randomly selected 100 papers published in the journal and looked at the papers for the sex of the cells. And then 20% of the papers used the male cells, 5% used female cells, and they did not specify for 75%, so it was not known 75%. So both of studies showed that 70-80% of the studies do not report sex of cells. So why the reporters or the researchers do not report the sex of cells they used in their experiments? Well, there can be several reasons, and maybe the sex of cells may be regarded irrelevant to their study because cell structures or basic functions tend to be similar, and they do not, it doesn't come to their mind whether they should compare male or female. Or it may be considered unnecessary because researchers really rely heavily on the previous papers, and as we said, 70-80% of the papers do not report the sex of cells. So it doesn't just, researchers do not consider they have to do it. But maybe researchers just do not know what is the sex of the cells they are using. So one of the researchers in our team, Dr. Suk-Kyung Lee, when we first discussed about gender in science, she realized that she never looked at the sex of the cells that she was using. She was doing cell experiments about stomach cancer. So she tried to find out the sex of her cells as well as what is the cells that other people are using. And she couldn't find the information. So we went in to look at how the sex description is available from the major vendors in the world. So we used the information from American Type Culture Collection, ATCC of the United States, and ACC of Europe, and then JRBC of Japan. And we published the results in American Journal of Cell Physiology. So cell lines and hybridomas, for the human cells, this is human cells, so about 20% of the cells do not have the sex description. And then about 70-80% have the description about their sex origin. But for the rat or mouse cell lines, most of them, we don't have the sex description. So the researchers do not have a way of getting the information. So we thought this is a serious problem because, as we just heard, now NIH is requiring all the researchers to balance their experiment for preclinical studies. And also it was in Europe, we also studied by Horizon 2020. So not only that the large global cell vendors do not provide information, they don't provide any search engine for the sex-based search. So there is no way for the researchers to find what, if they want female cells of certain disease type, there is no way they can find. They have to just go through one by one, and as it takes a much long time. So what we should do? So we decided to make up some tool for the researchers to use to balance this sex of the cell experiment. Now I want to briefly describe our center and the gender innovations activities in Korea. So the major role has been played by WISTED and COFST at the beginning, and now we have GISTER, Gendered Innovation Science and Technology Research. Now the first introduction was by Dr. Lee in 2013 when the European Commission published this case study book. So we worked through with the researchers of the Korea Federation of Women Science and Technology Associations until we had the Gender Summit 6 Asia Pacific in 2015. And the summit was quite successful, you agree? And we also were successful to get some budget from the government to expand this activity. So now we have established gender in science and technology research in 2016, about 2 years ago. So in GSTER, we have a policy research team, and it is led by Dr. Heskuli, many of you probably know. And she has been very successful to pursue several initiatives. And I'm not going to go into detail because of the time. And I think she'll perhaps present about some things in the future. She could not come at this meeting because of some personal reasons. And I lead the case study team, and we have 4 areas, and we have 18 case studies are now going on. And she's the one who, the first one in basic science is Dr. Lee, who worked a lot about the cell bank, cell database. And this is our website. We just opened the English website. And I hope if you have time, you can go through there. And so in the middle, you can find that there is a Gendered Innovations DB search. So we have developed literature DB, and then 4 kinds of database, what we call resource database. And these databases were started by the request of the reviewers of our project. And they said this is kind of new initiatives that should be applied to many fields of research. So if there is if other researchers who, besides our team, want to start their research with gender, they need some kind of resource to look for. So we compiled the data from the literature of the topics that our team is working, and also with some resources that we accumulated during our study. So today, you can use there are 2 DB search clicks that you can use, and today use the cell DB introduced to you, so that you can be familiarized and use if you are doing cell experiments. So to establish this cell database, we use the resources from 4 cell vendors, 3 vendors that we did for our paper: ATCC, ECACC, JRBC, and we included the Korea Cell Line Bank because we are in Korea. And so I think most of the researchers are buying their cells in one of those cell vendors. So for each cells they sell, and then they are described in their website. We we developed the software to enter all these informations about each species, and then sex and gender, and what categories they belong, and then disease model, and then tissue of origin, cell type, and then the vendor. So this is our database site. So total number of cells that are included in this database are 6,247 as of this month. So if we see the distribution, we have we have about 40-some percent of the cells know the sex origin, but about more than half we do not know the sex description. So in humans, we have more. We know more about their sex origin, but as I said, as we saw before, rat and mouse cells are very low compared to other animal models. So I will show you one example. If we are looking for human colon cancer cell line, and so we you can just click. These are the informations we have entered. So in each category you click. So in this case we said human cell lines, cancer model, and colon. So we have 134 cells in the cell bank available from the market. So for each cells, if you are interested in, you can click here, and also you can see the information, more detailed information from here. And now if we want to see the male cells, and there are 51 male cells among 134 or human colon cancer cell lines. And if you want to have female, you can just click the female, and there are 49. So if we have unknown, so how many are unknown? And we have 34, because out of 134. So it's about 25%. So 1/4 of human colon cancer cell lines, we don't know the sex origin. And you can also choose the specific cell type, for example, epithelial, and then among the female 49 cells, there were 41 epithelial cells. And you can also choose the vendor. If you are buying cells from ECACC, you can see what are the cells that they are selling for female epithelial human colon cancer cell line. Okay. You can also alternatively, if you know all the characteristics that you want to specify, you can just write in this box, and you will also get the same results. Okay. Do we have time? Finish? Okay. So this, I'll just go to the last page, but I'm just basically saying that this is a certain resource that if any researcher wants to find a certain sex of cells, they can come in, and then I hope this is used and then helpful to researchers in the worldwide to do a good experiment with both sex cells. And since this is a new database, I would very appreciate your suggestions or any opinions about how to make it more useful. Thank you very much.